Thank you so much for the kind introduction. I'm very happy to be here. So today I'm going to talk about the electrophysiology, the intracranial electrophysiology of speech perception and production. And I'm going to try to provide some temporal detail and how the time evolution of neural signals can inform current models. So I'm going to pick my favorite model of speech perception and production. And I'm going to focus on a few hallmarks of this model. So one is that there's an articulatory network in the inferior frontal gyrus that supports a production of speech, well known as Broca's area. The other is at some regions and some streams, you have to have a sensory motor interface that integrates what you're hearing and what you're saying. And lastly, the hemispheric asymmetry and the bilateral nature of spectrotemporal analysis during speech perception. That's basically the outline of what I'm going to be talking about. I'll start about the role of Broca's area in speech production, move on to sensory motor integration, and finish with a spectrotemporal view of hemispheric asymmetries in speech perception. Broca's aphasia, first described by Broca in the 19th century. And you see here the postmortem analysis of two of his first cases, case Tan and the second case Lulong. And you see that there's quite a large damage in the inferior frontal gyrus in the first case, but fairly limited infarcts and damage in pars epicularis of the second patient. But of course, these were the basis of the findings of Broca and the implication of Broca's area in speech production. So Nina Dronkers, who I had the honor to work with during my PhD, was able to get permission to actually take the original brain of the first patient, Tan, and actually the second, here we see the first, and image it in the MRI. And what you find there is, while there is evidence of damage to the inferior frontal gyrus or Broca's area, there's actually quite a larger involvement of underlying white matter tissue and other cortical structure beneath IFG. So it implicates something above and beyond just the IFG or the pars epicularis and triangularis. If you look at the early stimulation mapping data, so this is a paper from 1989, where Ogeman shows that indeed, when you stimulate intraoperatively to map out essential language sites, you see that these sites in black, when stimulated, the patient was unable to name objects. In M and S, you see the motor cortex responses, so movement in the hands when stimulated, or other muscles. And in white, areas that were cleared, so you stimulated and nothing happened, or maybe there was a partial hit. So indeed, you can stimulate IFG and get speech arrest and naming deficits. However, in other patients, you can map out IFG and be mostly cleared. So there's quite a intersubject variability in where these sites are. And the take home from this in the 80s was that naming errors, and then subsequently in other papers, speech arrest, happens outside the traditional regions of Wernicke and Broca, and especially for Broca, you don't necessarily get a deficit. In addition to that, if you look at some early papers dating back from the 70s, from Moore et al., you see that if you have damage that's limited to pars epicularis and triangularis, you do not develop a Broca's aphasia. You have a transient mutism that lasts throughout 10 weeks. And there you get subsequently much better and have an improvement where you're left with maybe an anomia and unable to produce and name words. But you make errors, but you still are able to fluently speak. So in addition to that, stimulation to Broca's area, as I just showed you, doesn't necessarily produce speech arrest. But at the time of this study, we had very limited data on the timing of activation. So we knew the areas involved in speech articulation, but the timing was clearly non-delineated. So during my PhD and my current lab work, we study and work closely with a patient population that has refractory epilepsy, untreatable by medication. And their last resort is really to go through a two-stage medical procedure. In the first, they have a grid of electrodes or probes directly on cortex or in cortex implanted for a 1-week period in order to monitor their epilepsy and the seizure focus, so the eye of the storm. After they mapped out these seizures, they go through another surgery where the area is resected and removed to treat the epilepsy. During this time, they're just waiting for seizures off medication, watching TV, talking to family, and we come in and consent the patients and ask them if they're willing to spare some time to doing some basic cognitive tasks for science instead of watching TV, for example. And most of the time, they're very happy to oblige. So I'll start by presenting a very simple task. So this is an auditory word repetition task. You hear house, you say house, etc. These are all words that are one syllable, so short words. So when you hear house, I'm going to zoom in on an STG electrode. You can analyze this electrode by electrode, and I'm showing you a time frequency representation. So the neural signals are averaged across trials and decomposed first by time in the x-axis in milliseconds and frequency in the y-axis in hertz, and the color scale represents the degree of power in those frequency ranges. And what's been shown over and over in the past 20 so years is that there is this high frequency activity you see here in red that correlates with the bold signal and correlates with underlying neural spiking activity. And there's a very robust signal of local cortical activity. And this allows us to track cortical activity with time and now space across the different electrodes. So there's robust activity in temporal, superior temporal gyrus or auditory cortex when you're hearing the word. So that makes sense. That's nice. But what happens further is while you're still hearing the word, you already have IFG coming online and is active. And precentral gyrus or motor cortex is not quite active at this point. So the key question is what happens when you're actually producing the word. So Broca's area was seen traditionally as an area that is the seat of articulation or coordinates articulators. However, when you actually produce the word around 1300 milliseconds, auditory cortex is suppressed, but there's actually no IFG activity. So all the activity in IFG is happening prior to articulation while planning the word and not during articulation per se. However, during articulation, you have precentral gyrus and motor cortex actually executing the command and coordinating the articulators. So if you ask a simple question, now not just with these words, short words, but we added tasks where there are longer words and there's a visual word reading task. So they read out loud the word. So different ways of producing a word, either short and long. And across these tasks, we ask when was peak activity. In blue, you see peak activity that happened prior to articulation. In red, peak activity that happened during or after articulation. You see the stark dissociation where all the IFG contacts are active prior to articulation. You can map out this peak activity locked to stimulus onset or locked to articulation onset. Everything here on the y-axis, and the red line is articulation onset. So below it happens prior to articulation. So in cyan are STG responses that happen early on, followed by green, which are IFG, all below the red line of articulation, and precentral gyrus, premotor, and motor cortices are active prior to, as well as during and after articulation. So a nice thing about this technique is that we have a very high signal to noise ratio. So we can look at single trial responses and you get a very robust picture of activation. So here you have stacked 5,000 trials across these different tasks across all of STG electrodes. And you see robust activity locked to perception. So 0 is stimulus presentation, x-axis is time, and black line are the sorted articulation response time. So when you're speaking, you get, again, this suppressed response. But what happens in IFG is pretty clear that activity closely follows STG, but the majority is prior to articulation, prior to this black line. And Precentral gyrus, you see activity that's prior to and during, but is really locked to reaction, locked to speech output onset. So there are a few things you can do with this robust SNR. You can do machine learning approaches that's been done very nicely shown by Eddie Chang's group and others. We focus in this study on effective connectivity. So the problem posed in effective connectivity is how do you move from a set of time-varying signals to understand what influenced each signal? How did information flow from one signal to the next and the third, etc. One way of doing this is cross-spectral coherence. So you basically are trying to find if the signals are oscillating together or are they off sync. And when you do that, you get a peek at the areas that they oscillate together are coherent. However, this is done in a pairwise manner. So you have a lot of spurious correlations. If you think 1 and 2 and 3 are correlated and 1 and 4 are correlated, you're going to think 3 and 4 are correlated as well, even though they're not. So you need to partial that out. You also have indirect true flows. So you think if 1 and 2 and 3, you have information flow, you're going to think maybe 1 and 3 is contributing information. And lastly, this is not directed. You don't know if information is going from 1 to 3 or 3 to 1. So we use a flavor of effective connectivity based on granular causality. It's called directed transfer functions. And we do it in a short time window approach. So every couple hundred milliseconds, we can see this connectivity and see what areas are sending information in what direction to other areas while partialing out other spurious connections. When we do that and we average across a region of interest, we see that early on locked to perception here in the x-axis in time, you have information flow or effective connectivity from STG onto IFG that peaks very early on, but closely follows feedback from IFG onto STG while you're still hearing the word. So this is the one syllable auditory repetition task. All the words are around 500 to 600 milliseconds. So while you're hearing the word house, you have STG to IFG closely followed by IFG to STG. And here on the right, you have lock to production articulation onset at 0. And when you move close to articulation, you see this here in blue, IFG is sending information to precentral gyrus up to, but stopping at articulation onset, closely followed by feedback and crosstalk from precentral gyrus back to Broca's area or IFG. So what this shows us is that during preparation and prior to articulation, there's this crosstalk between IFG with STG at an early time stage, closely followed by information flow back and forth between IFG and precentral gyrus. So while we can show that IFG is active prior to articulation and is doing information coordination, information flow across cortices, the last trick we had was to use a manipulation that we had in the stimuli. So these were all 3 phonemes, 1 syllable words, but some of them were real words like jazz, book, and love. However, we created pseudo words that were controlled for the phonotactic distribution. So the probability of ja appearing there, the probability of a vowel, the biphoneme probability of two pairs of phoneme happening together, all that was controlled for. So that meant that Jeet and Bav and Lud was a highly articulatorily pronunciation that is within your repertoire of articulation, such that unlike the word I said before, which I haven't ever said, this, and it was hard for me, this is a word I've never said before, Jeet. However, it has the same distribution, the same probabilities of jazz. So it's easy to pronounce, but it has a novel articulatory sequence. And the question we ask is, given a novel articulatory sequence, where is the heavy lifting? What region helps you create this novel articulatory plan? And what we find is in IFG, you get this divergence once you finish hearing the word, that for novel articulatory pseudo words, you get higher activity compared to higher frequency real words. And this is not the case in motor cortex that is executing the words. That for them, for motor cortex, it's the same representation because you have the same probabilities. Okay. So this work was done during my PhD, guided by Bob Knight, my PhD advisor, together with Nina Dronkers, Duke, and our clinical collaborators at Johns Hopkins. And I'd like to move from the story of Broca's area, where I hopefully showed you that that activity is prior to articulation and it doesn't coordinate the articulators, but sends information back and forth across cortices to create an articulatory plan executed by motor cortex. I'd like to move from there to the next part, which is sensory motor integration. And I want to go back to some of the data I showed you. So as you recall, I showed STG activity followed by IFG and precentral, but I kind of mentioned the suppression of STG. So why is auditory cortex suppressed during articulation? So this is a well-known phenomenon. So if you look at single units in marmoset monkeys, you see this large neuron spiking spontaneously. And in red, you see articulation onset, vocalization onset of the marmoset and conspecific vocalizations. And right prior to the vocalization and during vocalization, that auditory neuron is suppressed, while a neighboring neuron that's not as strong is not suppressed. But you get this very robust suppression of this auditory neuron during vocalization. And in humans, it was been shown in non-invasive EEG that when you hear vowels here in red, you get a robust N100 response. And when you speak the same vowels, you have a reduced N100. So there's suppression in single neurons, there's suppression in the EEG, or reduction in the N100 component. And this has really been demonstrated across different measuring modalities, and it's apparent across the animal kingdom. And you can see this both in EEG and ECoG up to intracellular recordings, where you see the influence of motor action on auditory perception. So this ECoG data is actually taken from this paper, J Neuroscience paper, where we showed not just the reduction in ERP like you do in non-invasive EEG, but we showed the distribution of suppression in high gamma. What we show there is that while the majority of STG is suppressed, there is quite a large variability, just like you see in the non-human primate literature. There's a variability in what electrodes are suppressed, which ones are enhanced, while a majority are suppressed. And you can also see this locked to single trial. So before I just showed you the average spectrogram, you have some jitter, but here there's a select electrode from every patient. And you see while there's a variable degree of suppression, that suppression is sustained across the different trials. So every time you hear and speak, that different electrode has a very specific topology of suppressed responses. So why is there suppression? So we know there is suppression across the animal kingdom. One hypothesis is you're suppressing auditory cortex in order to be more sensitive to your environment, to shifts in your environment. So in this pitch perturbation study in marmoset monkeys, Eliadis and all show that as you have a normalized suppression, so if you look at all the neurons that are suppressed and you look at the degree of suppression from very suppressed to not suppressed, you have a very strong correlation with a degree of enhancement, of neural enhancement, when you provide a pitch shift going up or down to semitones. So it seems like in the non-human primate auditory cortex, this suppression is there for you to be more sensitive to changes in your environment. The same paradigm was done in ECoG by Eddie Chang et al. In 2013. And while they find suppressed responses here in blue, and as well as enhanced responses to the pitch perturbation in red, they did not find a correlation between suppression and enhancement. So a postdoc in the lab, Muges Sertel, had the idea that perhaps one of the reasons we didn't find it in humans is because we use this pitch perturbation approach. And we were interested in more of the time domain versus the frequency domain. So we wanted to use a well-known paradigm called delayed auditory feedback. So we've all perhaps experienced this when we're wearing headphones and we hear the feedback from our own speech, and in long distance calls or Zoom going awry, we hear our own voice with a delay. And if it's a short delay, like 50 milliseconds, we're fine. But at longer delays, it can cause slurred speech, repeating syllables, slurred speech, as well as stutter-like phenomenon. So I'm going to show you a demonstration. I just gave an RA at the time a book, had them read the one page in the book, and did an online delayed feedback, either at 0 milliseconds or 200 milliseconds.
The dramatic growth in practical applications for machine learning. The dramatic growth in practical applications for machine learning.
So he's already smiling, something's going, it's hard for him to speak. He has some slurred speech, and it's prolonged for sure. So we wanted to do this in our epilepsy patient cohort because there's really no neural evidence, or at least invasive electrophysiology, describing the regions that are interacting in order to both deal with this delay as well as compensate. So we had one task where the patients did a visual word reading task of a multisyllabic word, 3 syllables like umbrella, and we pseudo-randomly interleave different delays. So one time they would read it and the feedback would be at 0, one time it would be 200 or 50 or 100, and we got a slight effect of articulation duration. However, it was most pronounced when we had them do a second block where they read out sentences. So sentences like the cereal was fortified with vitamins and nutrients. And as we introduced the delay, it of course got harder and harder for them, and the speech articulation duration became longer. In our preliminary data, when you looked at the first 6 patients, these are 23 pericilian STG electrodes, you see that as we introduced the delay, we got a sustained increase in auditory cortex in the amplitude. So the amplitude of these auditory neurons coded for the amount of delay that we introduced. So when there was a mismatch between what was planned to be heard versus what was actually heard, we get a very robust response. And the same thing happens during sentence reading. So it could be that this 200 milliseconds is happening longer because you spoke slower and you heard a longer stimulus. However, we get an increase in amplitude, both at the beginning and sustained increase coding for this change of feedback alteration. So again, in order to ask the question, is this delayed auditory feedback sensitivity, is that correlated with suppression? We first wanted to assess this effect of sensitivity to delayed feedback. So we take these peaks and we basically do a correlation or a regression line. So we get a measure of how sensitive every electrode was to this delay manipulation, so delayed feedback. And in a separate task for some of our patients, we also had an auditory word repetition task, so very similar to the tasks I showed you before, where when you just hear the word, you get this response in blue, but when you say the same word in the same electrode, you get a very significant reduction, and you can quantify that reduction. And then the question is, does this suppression correlate with sensitivity? And we found that indeed, in our delayed feedback task, we have a robust correlation where the degree of sensitivity correlates with the degree of suppression. So perhaps the reason why we have suppressed responses is in order for us to be able to be more sensitive to the environment. Okay, so we have since published this. This is on bioRxiv and we'll just accept it to publication. It should be out next month. So if we take the 15 patients that we had in the study and we look at responses, what was interesting is, again, for words, we get this increased amplitude response in STG as we increase the delay. But in motor cortex, for word reading, we didn't really get any differences. So both the dorsal and the ventral divisions started activity prior to speech onset and continued during, but we didn't see, this coding for our delay as we do in STG. However, what was surprising to us is during sentences where we have prolonged delay across the entire sentence, not only does STG code for that in a sustained manner, but so does dorsal precentral and ventral precentral. However, at very different timings. Now, it makes sense for motor cortex to have this divergence because as I introduce a delay, I cause a compensatory behavior where you speak more slowly. You speak more slowly, you provide your articulation is longer, so you're getting more of these red lines. So it's because of articulation. However, this is happening very early on in dorsal precentral. So we wanted to figure out to what degree precentral gyrus here is actually processing the feedback auditory error, or rather is it just because of delayed articulation? So in order to do that, we wanted to in some way match the articulation. So we have the no delay neural responses, and we have the 200 milliseconds durations, and we also have the spectrograms for those different articulations for those sentences. And you see that as you increase the delay, you get longer speech, slowed speech. So what we did is you would want to have a technique that can match 200 milliseconds and no delay in the spectrogram, so they're aligned. So we use dynamic time warping that it does exactly that. It takes the no delay in 20 milliseconds and morphs them into a space where the acoustics are matched. And once we have that morphed trajectory, we apply for those same sentences that we match no delay in 20 milliseconds, so the same words. After we morph them, we apply that in the neural data, and that should control for anything that's due to slower, longer articulations. And when you do that in ventral precentral gyrus, indeed, you see that this difference that we had before goes away because we controlled for the articulation duration. However, in dorsal precentral, while there's some of this later activity is diminished, we still get a very significant marked here in black amplitude response for 200 milliseconds versus no delay. So that would implicate dorsal precentral gyrus in actually processing the auditory error versus just the articulation duration. Reviewers weren't super happy about that, even though we thought we were being pretty clever with dynamic time warping. So we had to find more ways to basically show that we're controlling for articulation properly. So what we did is we matched articulations. We took words. So yeah, in general, you're slower, but there are words where the no delay in 20 milliseconds words, or in this case, sentences, there are sentences that weren't slower. So we took all the sentences that were kind of outliers in a sense that they matched during 20 milliseconds and no delay. So they were within 2 to 3 milliseconds of length. So we know they're matched for articulation, and we just analyzed those without doing any dynamic time warping. And again, when we match for articulation, we find that ventral precentral gyrus, we have no effect, no significant difference. However, we still get more activity for 200 versus no delay. So being in the 200 millisecond condition, those trials has a significant amplitude increase in dorsal precentral gyrus compared to no delay, even though it's the same sentence and it's the same length. Okay, so what does this actually mean? So if you look at it, I mean, it's quite robust. Like if you looked at this neural data and you didn't know what's going on prior to speech, it looks almost like auditory cortex. But you have this premotor buildup and you already start up very high up, but this divergence coding for an error is happening very early on. And if you look at the timing, you see that STG is significant starting around 80 milliseconds or so, very early, followed by dorsal precentral gyrus in a sustained manner. And then other regions coming online a bit later on. But it seems that the processing of dorsal precentral gyrus of this error, this feedback error, is happening very early on, akin to auditory cortex. It is not due to articulation per se. And it turns out that if you take the patients who actually had some passive listening data and you look at dorsal precentral gyrus activity, you see their responses there to just passive listening to speech. So there's quite a large literature to date of responses in motor cortex to passive listening. But what this, I would claim, says is that the reason you have passive listening, so auditory responses in motor cortex, placed in this region would be for it to store an auditory representation that can then be used during feedback and sensory motor integration, such that it can support the integration of speech production during the feedback of your own speech. So this work was led by Muge in the lab, and of course with our collaborative clinical team. And it will be out next month. And if you want to read it before that, it's on bioRxiv. Okay, so this leads me to the last part of my talk. So this will be about work I did in my postdoc, which is a framework for auditory hemispheric asymmetries with David Popel at downtown NYU. So evidence for hemispheric asymmetries goes back to the aphasia literature. And if you look at early 1970s studies, you see that many left hemisphere aphasics have deficits in processing temporal features like voice onset timing or temporal order. So there's a temporal processing deficit. And if you move on to the 80s, that's when, for the first time, Ross really established that there's a deficit in the right hemisphere in processing spectral features, such as prosody and being able to dissociate if I'm making a statement, am I asking a question, and as well as emotional content driven by the prosody. So there's a long line of literature showing electrophysiological evidence for an asymmetry in responses to temporal and spectral features, as well as neuroimaging studies supporting that as well. So what the models that try to explain this, there are several of them. I'm going to focus on two. One is from Zatorre that really tries to make a metaphor parallel between a time frequency decomposition. So in a time frequency decomposition, if you have a spectrogram, you can either have really good time resolution, but poor frequency resolution, or the inverse, where you can have really good frequency resolution, but then you have a poor time resolution. And because of this uncertainty principle, which is akin to the Heisenberg uncertainty principle, so it has a constant that if you go up in frequency resolution, you go down in temporal and vice versa, there's a theoretical constant. So because of this, perhaps what the two hemispheres are doing are trying to tackle the problem in parallel. So one is focusing on high temporal, one is focusing on high spectral, such that you can get your the cake and leave it whole, or eat the cake and leave it whole. So another model is from David Popel, who was my postdoc advisor, and it's very similar in nature, but it has to do with the windows of integration. So according to this model, the left hemisphere is integrating windows that are very short, around 25 milliseconds, and not integrating that much longer windows. And on the right hemisphere, you're chunking up or integrating very long windows that can capture prosody, et cetera, but not as much of these short windows. So these models are similar, but they have some distinct differences. So for example, the cortical computation in Popel's model is time integration. So your integration across time of the input. Where in Zatorre's model, there's this parallel between time frequency and time frequency resolution, but the actual underlying computation isn't really specified. If you look at spectral processing, it's explicit here in the high spectral resolution, but it's kind of underspecified in the asymmetric in time model, because it's implicit to the fact that when you have longer windows, you get better frequency resolution. So I really wanted to reframe this in modulation domain. So what is a modulation domain? You can think of a time frequency representation of speech, the spectrogram, time in the x-axis, and frequency in the y-axis as something that has slow temporal modulations or low temporal modulations, things that happen once or twice per second, or things that happen many times per second. You can also look at this on the spectral axis and see that you have things that are happening once or twice per octave, or you have things that happen many, many times per octave, right? So this is really a quantification of temporal and spectral resolution with this domain. And this view of temporal modulation and spectral modulation was really pioneered in Shiab Shamma's and Tunison's group, where they took speech and they borrowed from the analysis of in the visual domain and took the spectrograms and images and did this modulation domain. And when you take speech stimuli, take spectrograms and analyze the modulation domain, you have these distinct regions, first reported by Tapita Elliott in 2009, where you have regions that are critical for intelligibility and regions that are critical for pitch and gender identification. And when I first saw this, I was very struck by the fact that this separation is very similar to the neural separation and the hemispheric asymmetry literature. And I was hoping we could leverage this in order to create specific hypotheses. And you have two axes because you have to dissociate things that are sweeping down to the right versus things that are sweeping up to the right. So what is the new framing of the hypothesis? So the hypothesis is that the left hemisphere is again integrating temporal modulations, but it's integrating both short temporal modulations and long temporal modulations, or low frequency and high frequency temporal modulations, things that happen once or twice per cycle and things that happen many times per cycle. It still is integrating some spectral modulations, but a very limited set. And there's a core region that's shared with the right hemisphere, so low temporal modulations. However, it is limited in integration of higher temporal modulations, and it's integrating a wide range of spectral modulations. So this region here is shared by both hemispheres, but what distincts the two is the range of temporal and spectral modulations they're integrating. So in order to test this hypothesis, we have to manipulate stimuli. So we have a bunch of sentences from the TIMIT database, and you first create a time frequency representation of the sentence. You do a 2D FFT in order to get the modulation domain, and then you filter that. So if you do a filter of the spectrogram and you inverse FFT, you get a diluted version or a smeared version of your original spectrogram. So this spectrogram only has temporal modulations up to 2 Hertz, as you see here. And then we use a trick in order to create the waveform that corresponds to this spectrogram using convex projection. So I'm going to play to you what that sounds like. So that should be highly unintelligible.
Don't ask me to carry an oily rag like that.
And we can parametrically increase the amount of temporal modulations.
Don't ask me to carry an oily rag like that.
And if I primed you and I said the sentence was, don't ask me to carry an oily rag like that, you should be able to hear it more clearly.
Don't ask me to carry an oily rag like that. Don't ask me to carry an oily rag like that. Don't ask me to carry an oily rag like that.
Okay, so we then took many undergrads and we did some basic psychophysics at this very low resolution that was not published before then, where we had them listen to these sentences and write out the proportion of words correct. And what you see is there's a sharp cutoff around 5 Hertz where you do very poorly in intelligibility when you have up to 4 Hertz modulations. But as you increase them, you get a very sharp increase in the psychometric curve. And we were very careful not to prime them because you may have seen before, it's a very striking effect. And even by doing a second block, they get much better. And we report that in the manuscript. So we did another manipulation, which was not only did we play these low temporal modulation sentences, but the same sentence, in a separate group of participants, we played one level, maybe 3 Hertz in the right ear and a higher level, 4 Hertz on the left. And we counterbalanced and we took the first time they heard it and we asked, is there a difference if you got more information in the right ear or the left ear? Null hypothesis would say, there should be no difference in which ear you got more information. However, if there's an asymmetry in cortical processing or even subcortical, then perhaps you get asymmetric responses. And we basically replicated, the classic Kimura dichotic effect with syllables and consonants, but we did it with temporal modulation. So we see that there's a significant right ear advantage implicating the left hemisphere in processing temporal modulations. So if you give more temporal modulations, the right ear, the left hemisphere, can do more with that for intelligibility. And then we wanted to test this neurally, so we used our favorite neuroimaging technique, which was MEG. And we correlated now. We did the same tasks in the MEG, but we ask what is the neural power? So this is 0.1 to 8 hertz. So neural power as you hear sentences, and every sentence has more and more temporal modulations. And as you see, as I increase the temporal modulations, we get increase in neural power, which starts to diverge very early on once you hear the sentence, and by the end of the sentence, it converges. So it's really specific to hearing the sentence, this effect. If you quantify this effect, you get significant correlations across patients and sensors marked via permutation here in black. And you can also look at the topography. So it starts out at the first second or so to be very left hemisphere, or to be left hemisphere lateralized, and it becomes more and more left hemisphere lateralized as you go and advance through the sentence. So the task again was just to mark 1, 2, 3, or 4 how intelligible the sentence was. And then we want to do the same thing in the frequency spectral modulation axis. So we do a low pass filter on the frequency, and that basically smears it across frequency. And the question I'm going to ask now is it a male or female speaker?
Allow each child to have a nice pop. Allow each child to have a nice pop. Allow each child to have a nice pop.
So as we increase it, and of course we did the same thing with the participants in the lab, as we increase the spectral modulations, we get a very sharp psychometric curve that as I increase information, you get much better at dissociating male and female. And behaviorally, you always categorize this robotic type low spectral modulation voice as male. And as you have enough content, you get better at identifying the female speakers. So again, we did this in the MEG, and as I increase the spectral modulations, I'm getting increased neural responses. And they're significantly correlated, however, to a lesser degree than the previous task. And we start out with a bilateral distribution that becomes a bit more right hemisphere lateralized as we continue. And if we do it across the task and correct for multiple comparisons, what we get is, a more left lateralized distribution of correlations for the temporal modulation, and for correlations with spectral modulations are more right lateralized. So the last thing we did is we collaborated with neurosurgeon at LIJ, Ashmita, who had, instead of using grids, as I showed you before, he uses this approach, which is stereo, which has become more and more popular these days in North America, but has been done in Europe, since the 60s with Talirach. And in some cases, he had rare coverage of bilateral auditory cortex. And in this case, that's exactly what we see. These are 2 electrodes, and we did the same task, though shorter, with a stereo EEG patient. And you can see the same neural power within one patient, 1 electrode, and we get the same type of correlations with temporal modulations on the left electrode. And on the right electrode, we also get significant correlations, but again, the effect size is a bit smaller. So we have many more controls and information in the supplementary material. We show that on the left, this happens both in low frequency neural power as well as high frequency, high gamma, as I showed you before. Same thing with the right. We show the distribution of all these correlations across cortex and high gamma. And if you perhaps had an issue with intelligibility in the task being confounded with temporal modulation, which it is, we reran the same sentences, but reversed the speech. So we still have the same temporal modulation filter, but they're all unintelligible. And indeed, you still get significant left hemisphere correlations as a function of temporal modulations, even though you're just listening to passive listening with attentional control to reverse speech. So this was done in my postdoc with David Popel, with collaboration with Ashmita, as well as the clinical team here at NYU, where we got more grid patients and coverage for the paper. So I hope the take home was that Broca's area coordinates activity prior to articulation and sends information across cortices to create a novel articulatory sequencing plan. That dorsal precentral gyrus is processing auditory information specifically to have representations to guide feedback. And that temporal and spectral modulations is a good space to look at hemispheric asymmetries, and it definitely drives asymmetric responses. And this is the current lab, and I am now open to questions. And of course, I'd like to thank my funding agencies, without which I couldn't get this done.
Thank you very much, Adeen. As we always say, you should be hearing the virtual applause from your audience, but they're all muted, so you're not hearing it. Thanks a lot for your talk. That was great. We have questions in the chat box. I'm just going to read those out. First question is from Kirana Tsapkini. Very interesting results. I was not clear what the task was when STG, IFG, and then motor cortex got involved at the different time windows. For example, is IFG involved when the task is not repetition, such as when the task is explicitly just to listen to words, but not to repeat? I'm trying to get to two questions. What is the role of the task for the activation of each area, and whether there are any control processes that would be assigned to IFG or other areas if planning for production should be restrained? Finally, do you see different parts of IFG, for example, triangularis or opercularis for words and pseudo words?
Okay, those are all great questions. Let me answer them one by one to the degree of what I can answer and what I cannot answer. So the task was auditory word repetition. So the data I showed you was you hear a word, then you say a word. So indeed, it's within this task. It also included visual word reading. So you see the word and you say it. So regardless of that, it came online in a similar manner. To what degree is this perceptual processing of the stimuli versus articulatory planning? We did have, this is actually currently unpublished and it's part of my current grant. We did have passive listening responses in IFG. They were not as strong as the pre-articulatory processes. So if you just mapped IFG neural responses, it would be very high when you're preparing for articulation within 20 to 40 milliseconds, and you still get activity for passive listening, but not as much. However, I mean, it came out in the end because it was very confusing for reviewers to explain to what degree is that perceptual, or is that, you know, some kind of automatic planning or subvocal rehearsal? So you actually need to do many more controls in order to tease those apart. And that's part of the current work, which is why we also had some passive listening data for Muget's project as a control. Now, for the next question about pars opercularis and triangularis, for that study at the time, I had 7, 8 patients. I was very limited to that parcellation. We didn't really find significant responses, or I didn't really feel comfortable making that statement with insufficient coverage. However, right now in the lab, we actually have, some of the grants were developed in order to answer that, the differences between pars opercularis, triangularis, the distribution, etc. Suffice to say that we have better coverage here, and we do have publications coming out that show differences in pars opercularis and triangularis. And I believe that this pre-articulatory response is more prominent in pars opercularis, but you have to stay tuned to more publications for that.
Great, thank you. I had a follow-up, but I do notice that part of that was actually part of Kirana's question as well. Still though, in the repetition task, showing the timeline of activity in STG, IFG, and precentral gyrus, do you know if there would be an effect of lexicality? If you talked about it, then I missed that. So that is, would IFG be similarly active during non-word repetition, and could that distinguish between a role in articulatory planning versus modulation of lexical access and comprehension?
Right. I mean, So right now the lexical item of a non-word is really, it's zero frequency versus high frequency. We did look, I looked at other data. I looked for effects of, within real words of word frequency and lexical axis. And I think there's some reports out there. In my data, I didn't really see significant activity of word frequency in IFG. So maybe I didn't look at it correctly, but I didn't really find that. And given I didn't find that, the most prominent effect was between the real words and pseudo words that were controlled for. So we interpreted that as articulatory sequencing.
Yeah, makes sense. Thank you. While we're waiting for other questions, I did have another one as well, which I typed in, so I'll just go ahead. To what extent do you think these subtle temporal sensitivities that you talked about in the second part of your talk in the brain may be neuroplastic? So that is sensitivity to particular frequencies or spectral modulations dependent on specific cell types, which may then be limited to certain regions?
Yeah, I mean, I had, I think, a similar question once in a previous talk. I can't really speak to, cell types, right? Because we don't have that granular resolution. So I don't really know. However, I will say that, this task has effects that are, you adapt to, right? So people who stutter and people with Parkinson's disease, you actually would do a delayed auditory feedback online, maybe with pitch perturbation, to help their fluency as a reverse effect with people who stutter, but they adapt to it after a while. And so the question of adaptation, I think, is very important, of how we adapt over time. And that must be, some kind of plastic process, to what degree we can ascertain the correlation of plasticity with cell types and then correlate with STG location is very hard to say, besides maybe using, something like Amunds's atlas to try to parcellate probabilistically. I will say that in our task, we looked at adaptation and differences in behavior across, in some patients we had more blocks, across blocks, and we didn't really see adaptation within, a 20-30 minute window. So it could be that this adaptation process is much longer, to a degree that we couldn't assay.
Yeah, I'm asking, of course, from the perspective of someone doing stroke research too, right? So what if those areas are gone, which is the case in some of our patients, or severely damaged? What are the options of retaining or rehabilitating some of the functions that they served?
So that's a great question. I'm gonna answer it in a slightly different way, in a sense that the third part of hemispheric asymmetries, part of the design was such that I wanted to really fine tune the sensitivities of left and right, because perhaps if you know that space and you can modulate and see what the sensitivities are, perhaps you can create training mechanisms for perception that you can retrain the right hemisphere to do what the left hemisphere is good at. So it's actually designed in that in mind. And if we go to the auditory feedback project, I don't have this data, but there's data in Nature Communications. I forget the author right now, but where they basically show an fMRI, a temporal kind of compression of speech versus pitch perturbation. And there they show left versus right preference for that. So it could be that you could do a pitch perturbation versus a time alteration, feedback alteration, and kind of do a retraining of one hemisphere versus the other in some manner. I think we're very far from that at the moment. And even if you're successful, to what degree that would, then generalize to language abilities, right, or higher order perceptual abilities. But I will say that in our data, we do have left and right coverage in Muguet's paper, and we don't have as good a coverage of the right, but we didn't really see differences in our task.
Thank you. Thanks so much. I think we've come to the, there is another question from Kirana Zapkini, who also says, wonderful talk, thank you. Could you speculate a little more on the dorsal precentral gyrus? Would you say it is a control area for sentence comprehension?
Yeah, it's hard. I mean, it's always easy to speculate, but I like to speculate out of what I know. So this is how I think about it. I think that there's I've seen across my graduate career a lot of responses and auditory responses in motor cortex, and there's a lot of signatures of that in ECoG data. Spectral temporal receptive fields in speech motor cortex from Eddie Chang's group, Chung et al. And eLife. And I just showed you a bunch of passive listening responses, and we see this very thoroughly. So the question is, why do you have an auditory representation, an auditory evoked response in motor cortex? And to that degree, I speculate, and I think we have some evidence towards that it's there in order to keep a representation such that it can better integrate when you're speaking and have an auditory space target, kind of like in the DIVA model, we have an auditory space and somewhat the sensory space. I would say they need to add an auditory space for dorsoprecentral gyrus so you can do very fast predictions and lookups and changes between what you're saying and what you heard. And you don't have to go back and forth from SDG. You have kind of a template in motor cortex to make it happen faster. That would be my speculation. To move forward to sentence processing per se, I mean, I think the difference between sentences and word in our task is probably also due to a block effect. So there's a block of words and a block of sentences because the timing of divergence in sentences already happened in words. You got to it, but you didn't diverge. So so it's either the block effect, or for words, there are 3 syllables. You already have that in the buffer working memory phonological store, such that you don't need to compensate because you're not hitting disturbance already. It's already there. You feed forward, move it into articulation. Yeah, you get delayed feedback, but it doesn't interfere. When does it interfere? When you keep on planning. It's not a one-time plan. You need to plan the next word and the next syllables, and then you get interference. So that's how I see it. So not necessarily sentences per se, but longer planning.
Thank you so much. And that really does bring us to the end of your presentation. Thanks, Adeen.
Thank you.
Good to see you. Thanks for attending, audience, and we're looking forward to seeing all of you in 2 weeks' time for Greg Hickok's talk in the series. Until then, safe travel.